I will be discussing here the first of the three major steps of the central dogma, which is replication. So, first, if you've watched my overview of the central dogma, you may recognize this diagram right here. And replication is basically this process wherein we start from DNA and then we end with DNA. Remember, just like what I told in that overview, the reason why we need to produce more copies of the DNA is to prepare cells containing that DNA for cell division. Okay, and sometimes, therefore, we could say that replication is called DNA directed DNA synthesis because you start with DNA, so that's the director, and you synthesize the final product, which is also DNA. So DNA directed DNA synthesis. And of course, we should imagine that it depends on whether it's a prokaryotic or eukaryotic cell that we're looking at to imagine where this happens. If it's a prokaryotic process, most likely this is in the cytoplasm. If it's a eukaryotic cell we're looking at, replication is done inside the nucleus. In either case, we could imagine this starting point. So it doesn't matter if we have a prokaryotic and eukaryotic example, it's the same. So imagine I have here a DNA double strand, which again, as you could see, follows the properties we've discussed before. That first, we have here a helical arrangement. They are coiled in some degree, and that the two strands are anti-parallel, such that the blue here has the direction 3 prime to 5 prime, and then the red one here is 5 prime to 3 prime. Now, in replication, we have to recognize that in any DNA strand, there's a part of it that's it's kind of predetermined already. Okay, in every DNA strand, that this is what we can call the origin. So of course, I just assume that in my drawing here, let's just say that this is the origin, the one with the a white circle, and that's the reason why we call it the origin is that that's where we start things off. So, to start things off, once we recognize the origin of replication, we need to call out the enzyme helicase, which from its name helicase will try to break the helical arrangement, this kind of, spiral arrangement of DNA. So you could imagine as if this figure right here is the helicase enzyme, and you could imagine this thing right here going to the origin and trying to break apart the connection between the red and the blue strands. And as it does that, it slowly opens this up into some kind of hole right here or cavity. And of course, you should imagine that this is a dynamic process. That as my helicase goes further in this direction, so as if I'm going from left to right, you could just imagine that this cavity or this gap right here is continuously getting larger and larger. This gap right here or this cavity right here is often called the replication bubble, okay? Because the cavity is kind of circular, so we call it a bubble. Or sometimes we call this area right here as the replication fork, okay? Because here we could see that the two strands are forking out at the end where the helicase is moving, you know, or on the direction where the helicase is moving. And notice that the direction of helicase is going farther and farther from the origin. So at any given point, the replication bubble will have two ends. At one end, we have the origin. At one point, we have the helicase, wherever the helicase is at the moment. And again, as I mentioned, this helicase is just going farther and farther and farther and farther from the origin. And the replication bubble, if you could just imagine this thing moving, it's just gonna be larger and larger and larger. Probably until all of these strands, red and blue, completely separate. Now, once we do have this replication bubble being formed by the helicase enzyme and again becoming larger and larger, we have to recognize the fact that at other points of the DNA strand, there's continuously building up of tension. Okay, so it's as if as my bubble here or this gap right here becomes larger, you could just imagine that the rest of the things right here are being more compact. They're becoming disorderly, and there's a lot of pressure or torsional strain building up among the nucleotides here. And the worst case scenario is if the pressure on these things or these parts build up too much, this may break apart. And there's nothing to work with. There's nothing to replicate in a DNA that has been broken apart. So, in order to relieve the stress or the strain being enforced by the replication bubble on other parts of the strand, like here and here, we have to have the help of enzymes called topoisomerases. Though in eukaryotes, we often call these enzymes as DNA gyrases, but as far as what they do, these two words pretty much do the same thing. Okay, probably just have differences in the finer details, but yeah, the purpose of the topoisomerase and the DNA gyrase are to stabilize the other parts of the strand where we expect the pressure and the torsional strain to build up. Now, another thing is that, the moment that we have this replication bubble, it is quite tempting for these two strands to reunite. To reunite. Remember, I mean, if this base right here and this base right here at one point become a little close to each other, they might eventually lock again, and that would be sad because remember the purpose of the helicase is to separate these two strands, and it would be useless if I have these two separated regions coming back together when we don't want them to. So in order to prevent them from reuniting back together, usually we recognize the presence of proteins that try to, you can imagine these as like nails or s- s- uh, stapler wires that once these things are attached to our single strands, they prevent these two strands from reuniting, and these proteins are often called single strand binding proteins. Single strand binding proteins, because their purpose is to basically bind to the single strands and prevent them from becoming a double strand again. So yeah, so you notice there's a lot of emphasis on trying to stabilize the replication bubble because again, this is not the normal way that we find DNA. We actually forced the bubble to open, and so it makes sense that to compromise for us forcing this thing open, we have things like topoisomerases or single strand binding proteins. Now, okay, this actually contributes or constitutes most of what we call the initiation phase of replication. At this point, we've just set everything up, but as you notice, we're still not making anything yet. So, in the next step, which is the elongation phase, we are bound to see additional strands or additional nucleotides being added. And here, I'm going to use the color yellow to indicate any new strand being synthesized. Okay? So anyway, after we have the replication bubble, we need the next enzyme, which is primase. And the primase adds a so-called RNA primer, which I will color code as green. So if based on its name, the meaning of the primer or the purpose of the primer is to start the elongation process. For some reason, kind of complicated to mention here, but let's just say that for some reason, it is not possible to create an entirely new DNA strand from nothing. Okay, we have to have some kind of initial product that initial thing is what we try to build upon, and that initial thing is actually the RNA primer. So as you see here, I tried to now zoom in, particularly on the blue strand. I put my RNA primer here at the very start. Okay, and once I have my primer ready to go, the next enzyme that we need is called DNA polymerase, which I will denote as an orange square. Okay. And the purpose of the DNA polymerase is to basically make the next DNA strand, which is actually this yellow thing right here. So remember, there's no yellow a while ago. It's just red and blue. So the fact that we're now seeing yellow strands here means that this is the new one we are making. And it's thanks to DNA polymerase. Now, do note that the elongation process will always go from the direction 5' to 3' of the new strand. For example, remember, the color blue strand right here has the direction 3' at the left and then going 5' as I go to the right. Now, if that is the case that, my blue one is 3' to 5' and the yellow strand should be anti-parallel, shouldn't that mean that the yellow strand starts here at the 5', right? Blue here is 3' to 5'. So what's the opposite of 3 prime? 5 prime. And what's the opposite of 5 prime? Is 3 prime. So isn't that true? That here, you notice my polymerase is here. So my direction is in this direction here. And are we following 5 prime to 3 prime of this new strand? Yes. Okay. So relatively easy to understand at this point. And of course, we can't pull something out of thin air. So remember, these nucleotides must have come from somewhere. So we assume at this point that we have deoxynucleotide phosphates like deoxy ATP, deoxy GTP, deoxy CTP out there somewhere. And then the DNA polymerase just, grabs them and then tries to place them on this strand. And that is by the help also of the magnesium ion, which kind of stabilizes the formation of these bonds to each other. Okay, now there's another problem that we have to deal with here. It might be easy to understand that, if I have here the origin and then I have here the direction 5 prime to 3 prime, we have nothing to worry about at the upper region because if this is the origin and then helicase is going in this direction and then here I'm going in the same direction and I'm following 5 prime to 3 prime, there's no problem. So of course you may ask, what could be the problem? And the problem is this. What if I note the synthesis of the DNA below? Surely, if I'm making a yellow strand for the blue one, I should be also making a yellow one for the red. But if I follow the same direction as helicase at the red one, remember, left to right, what would be if I follow this at the bottom? What direction would my yellow strand be doing if I go from left to right? Now, analyze it. Since your red strand right here is in the direction 5 prime to 3 prime, wouldn't that mean that if go at the same direction, my yellow strand would be anti-parallel? So if this is 5 prime, then this direction right here for the yellow is 3 prime. And then if I go to the right, that would be 5 prime. So if I'm going to follow the same left to right direction for the bottom part, I would be making my strand or I would be elongating my strand in the direction 5 prime to 3 prime. And that's a problem. Why? I just said a while ago that elongation always, always, always follows 5 prime to 3 prime. And by going left to right also for the yellow strand here below, I violate that direction. I'm going 3 prime to 5 prime if I go left to right. Therefore, it is not possible to go from left to right at this red strand below. So what could be the only resolution? Well, we have to admit that we cannot go against this. So if this is if 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 a while ago I said that the yellow strand below has this area as the 3 prime end, the left one, and then the right one is the 5 prime end, and the direction of elongation is always 5 to 3, then we have no choice but to go backward, as in right to left. So notice where my polymerase is right here. Why is it right here? That is because we admitted that we have no choice but to first put the primer somewhere in the middle, and then because we need to follow 5 prime to 3 prime, we go here. And thus, my polymerase is actually going to the left as far as the red strand is concerned. Okay, and I will be discussing this in, I think, the next slide, that eventually, once I stop somewhere here, I'm gonna have to add another primer at the right, and then I'm gonna have to go my way backward again. And then there's something that we're gonna do here to link these two things together. But the point is this: since here we're actually making, pieces of DNA, but they're not completely, as smooth as the one here, because here it's just continuous. Add another one, add another one, add another, add another, add another. We're not violating any direction. But here at the bottom, it's like I'm gonna go backward, 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 backward. If there's a dead end, I have to go to the right side, add the primer, and then go backward, go backward. There will be a point wherein I have one set of nucleotides, a short one, and then another set of nucleotides. So they're fragmented, they're separated. And these fragments are what we call as Okazaki fragments. And of course, being fragments, I have to discuss later how these fragments are connected eventually. But do know that in one strand, there is no Okazaki fragment, but in the one strand here, we have. So now I'm moving to another slide just to show you how the Okazaki fragments are connected. Okay, so imagine, notice I'm just gonna use the red one here because I just, based on my drawing a while ago, it's the red one which has the fragments, not the blue one. So imagine that this is one Okazaki fragment right here, and it's as if we reached a dead end because this is the origin. So we have to find something. Of course, we should just imagine that the cell knows, okay, where to start here. Let's just say the cell decides, hey, I should place a primer here. And then, of course, since it's 5 prime to 3 prime here, I need to go backward from right to left. So, once again, DNA polymerase adds nucleotides one by one by one. So it's elongating right here. And then this is now a dead end because this is the first fragment, and then this is the second fragment. And we have to accomplish the connection of these two fragments. So how does that happen? First, what actually happens is that number one, we have an enzyme that removes this primer and replaces this primer with a DNA component. Now, remember this: replication means the synthesis of DNA. So supposedly, everything we're putting here are deoxynucleotides. But look, isn't it a while ago I told you, and it's written here, that the primer is made up of RNA? So technically speaking, although the primer is important to kick off the elongation process, it's RNA at the end of the day. It's not DNA. So that has to go away later on. And so there should be an enzyme that removes that and replaces it, RNA. So ribonucleotides would be replaced by the deoxyribonucleotides, thus the color yellow ones. And it's actually DNA polymerase which does that also. So the first thing that should happen here is the replacement of the primer. Second, okay, we now see that uh, we now have a gap between the first Okazaki and the second Okazaki fragment. And so this has to be sealed uh, by a special enzyme. And the enzyme that does that is called DNA ligase. Okay, so in that case, you now see that the two Okazaki fragments are connected. This is now one long strand. And this will happen again and again and again. So if you just want to see that happen once again, so this is one big Okazaki fragment already. Then I could make another one. And then just like a while ago, then a polymerase will elongate this in this direction. So here is my kind of dead end because I have the primer. So first, just like what I said a while ago, replace this with the proper deoxynucleotides, and then by the help of DNA ligase, you seal it. Now, what I usually imagine when connecting Okazaki fragments is like uh, back stitching, because like you put a primer, you go backwards, and then you add another primer, and then you go backwards, and then you add another primer, and you go backwards. It's like stitching, right? So that's basically the movement that is being done on the strand where Okazaki fragments exist, or on that strand where our direction of elongation is actually going towards the origin. Okay, because right, we're going towards the origin instead of away from it. So you will notice that there are actually a lot of things that DNA polymerase does, because I just said a while ago, it is the polymerase which elongates the yellow parts, and it's also the polymerase which removes the primer. So what gives? That is because DNA polymerase has several functions. For example, the one I told you a while ago, wherein we continuously add nucleotides one by one, is the 5' to 3' polymerase function. So we say that this function is that function of DNA polymerase that actually does the elongation process. However, it's also it's also DNA polymerase which removes the primer, but that's a different function. That is our 5' to 3' exonuclease, which does the primer replacement. So it's the polymerase which does the elongation, it's the exonucleus one that removes the primer or replaces the green ones with the yellow ones a while ago. But also, do note that we have the so-called 3' to 5' exonuclease. And it does what we call the quote-unquote proofreading function. What's that? What's proofreading? Proofreading is basically the way that DNA polymerase senses mistakes. The reality is that replication involves addition of thousands of nucleotides per second. In other words, it's very fast. And it is possible that in our own cells, they make mistakes, no, honest mistakes, because it's bound to happen. So it kind of saying it's a spontaneous event. It just happens. Our cells make mistakes. But of course, if our cells make mistakes in making our DNA, those mistakes have to be corrected right away. It's as if you type so fast and then you type a word wrong. There's an extra letter or there's a lacking letter, and you correct that. And isn't that what we do when we proofread? So technically speaking, the 3' to 5' exonuclease removes wrong bases whenever there are wrong bases. So, for example, if it's supposed to be adenine and then the polymerase mistakenly adds cytosine, the proofreading function will recognize that, remove the wrong one, and then, of course, add the correct one. Okay, it's as if this is the wrong spelling, this is the correct spelling instead. So that's what the exonuclease does. So notice the DNA polymerase is kind of a very busy enzyme because it has three very important functions. Now, so far, how could we imagine our replication? Because there's a lot of things going on. So first, for the blue strand, or the strand wherein our elongation is going away, right, away from the origin, or you could say in this strand wherein my elongation is to the direction of the primer, it's a continuous strand, right? The elongation here is continuous because it just goes on and on and on and on and on and on without interruption, without Okazaki fragments. And so the continuous strand is sometimes called the leading strand because it's the faster one. There's no interruption. So theoretically speaking, this would be done, or all of the elongation going on here would be done faster than the other strand. On the contrary, the other strand where we find the Okazaki fragments, okay, is said to be a discontinuous strand because the moment you form one fragment, you stop there and then you jump at one point and then you go back and there are gaps in the middle before you seal them with ligase. So it's not a continuous process, right? So that's why we call it a discontinuous. And on the discontinuous strand, wherein you find the Okazaki fragments, you expect that this, you know, the synthesis of the DNA would be slower here because there are a lot of pauses in between. So we call this as the lagging strand because it lags behind. Now, you also notice that since on one strand, the replication is going here, and then on one strand, the replication is going on the other side, it's as if replication happens in two opposite directions. And that's why we say that replication is a bidirectional process, okay? Wherein one half goes in one direction and then the other goes in another direction. You could also say that this is a semi-discontinuous process because, as you could see, half of this process is a continuous one and half of it is a discontinuous one. Now, at the end of the day, from remember, we started with blue and red only. Okay, we could say that this is the parent strand. Our parent strand is only blue and red. But at the end of the day, imagine as if these green things are gone. We're supposed to replace the primers, right? And then the yellow ones are all that remain. What are the colors that we would have at the end? One DNA would be blue and yellow, just like this, and then the other DNA would be yellow and red. We could call these as the daughter strands because they are the ones that come from the parent. So, in summary, you could actually say that in replication, one parent strand is converted to two daughter strands. And notice that in every daughter strand, half of the daughter strand comes from the original parent and half of it is newly made. And since half of it comes from the original parent or is conserved or is, passed on from the parent, we could say that replication is a semi-conservative process, okay? That one half of the parent goes to both of the daughter strands such that each of these two would have a portion of the original one. So, remember these words. These are things that we could use to describe replication: bidirectional, semi-discontinuous, and semi-conservative. Oh, and one more word to indicate that our process of replication has proofreading. It's very, very accurate. Our cells can detect any mistakes and correct them anytime. We also say that in replication there is a high fidelity. And remember, this is important. It should be very important that there's high fidelity or accuracy in replication. Why? Imagine if you replicate the wrong DNA, you will eventually produce the wrong RNA and potentially the wrong protein. And so, among the three processes, the most disastrous to get wrong is the first one. Because you get the first one wrong, everything will be affected. So, as compared to transcription, it is well known that replication has a much higher requirement for fidelity.